Alrighty, ladies and gentlemen, thank you for coming out tonight. I know it's late, so I'm going to try to keep this lively, and if I don't, well, then Foster will boom me off the stage. As he said, I'm doing tissue culture techniques in Virginia bluebells and Venus flytraps. This is in Dr. David K. Foster's lab with the Department of Biological Sciences here at Messiah. Theory introduction, and this is a lot, so I'm going to try to keep it really simple here. But there are essentially 5 stages to tissue culture, four that are tissue culture, and then the 5th, which is the end-use product here. Stage 0 is a big focus of what I've done this semester, which is selecting the plants and selecting where I'm going to use them from. Stage 1 deals with initiating roots and shoots, just getting them from that stage 0 into a tissue culture. Stage 2 is multiplying them over and over and over until you get the amount that you want. Stage 3 is prepping them to go into a non-sterile area. Step 4 is the finalization of that preparation in moving it to an end use here. So, why are we using fly traps? They're a great model organism. We've had great success in our lab with them and with multiplying them over and over again. And we have a ton of them, so it's really easy for me to be able to practice my technique on them. What's the problem with bluebells? There's 2 problems that we found contribute to it. First off is this cold tolerance idea. We think that there's something that happens when they get cold that shuts down their ability to be cultured. And since we take them from a greenhouse bed and put them in Percival chambers at 4 degrees Celsius, well, that's a problem. Because I don't know if you've noticed, but 4 degrees Celsius is pretty cold. The other one is preformed flower buds. Sometimes we think that there might be some mechanism behind the flower bud already forming, being later in the life of the plant, that causes it to fail in tissue culture. So there's our failure. Our research goals here is to look at the link between cold tolerance, preformed flower buds, and bluebells in tissue culture. Is it purely environmental? Is it those 2 things that I mentioned? Is there a genetic component? We'll find out. So this semester specifically, it's technique, technique, technique. We really need to figure out how can I as a scientist learn how to do these things? Because I haven't had a plant propagation course, and I have not had a course in tissue culture. So this was my crash course in tissue culture, and it was a doozy. The other thing we actually did this afternoon, about 3 hours ago. We began initiating primary tissue culture of some etiolated tissue that's been grown in the dark. And then preparing stage 0 bluebells was my overall work with the bluebells specifically this semester, taking them from the beds, moving them to the Percival chambers, getting them ready. So here are the bluebells. There is a picture of them from about a month ago in the Percival scientific chambers. We're monitoring their water usage, making sure that they are just surviving and thriving, hopefully. They should not pop above soil. If they pop above soil, I'm doing something wrong, because we're trying to keep them in there until about January, February, that time. Media preparation. This was the first step to tissue culture, and this was one of the things that I spent the most time myself doing. Our media consists of distilled water, which is just your aqueous base for everything. Your Marashagi and Scooge basal salt mixture that has macro and micronutrients. It's what's going to help keep the plant alive, not the animal. Inositol is a hormone that we use in it. So is indole 3-butyric acid. Sucrose is sort of like the plant's food. It's the energy source for things. And then agar is just the structural support, and it's what is allowing the plant to grow in a tissue culture. It's that jelly stuff that you see in Petri dishes or in tissue culture tubes. Stage 2 tissue transfers was what I spent most of my semester doing, perfecting, learning this technique. Because if I can't do this successfully, there's no way I'm going to tackle stage 1 transfers, and there's no way we're going to be able to multiply the plants unless we bring in someone else to do it, which would defeat the whole purpose of me doing the research. So stage 2 tissue transfers is pretty complicated, but it comes down to this. First, we're going to prep our sterile environment. So this involves turning on a UV light, spraying the entire area down with alcohol, and waiting 30 minutes. Why am I waiting 30 minutes? Well, I do like to keep my retinas, and UV light damages those. So yeah, you can see where I'm going with that. Following that, I also put my tools into the Germinator 500 there, which is just a bead sterilizer that's really hot. We dip it in 95% ethanol and place it in those. It's sort of like if you had Bio 170 here, where you would dip it in alcohol and light it on fire. It's the same thing, just with less fire. And then we take the plants here. We're going to take a full vial, which actually looks a little more full than this. This is one of my successful transfers. But you take a full vial of them, you pop it out with a really long set of forceps. You're going to place it onto a Petri dish. It does not close completely, so you take a scalpel, cut it into 4 sections, place each of those on its separate Petri dish, and then further segment those and place them into the tissue culture, all the while trying to maintain sterile technique. Can you see where the problem is? The sterile technique part. Like I said, it's problematic. If you fail in sterile technique, you get something like this, and this does not look like Bluebells to me, folks. It's not. This is some kind of bacterial mold, something that contaminated it and grew, because when you give it plenty of food and awesome conditions to grow, of course it's going to multiply like this. So I did 3 sets of tissue transfers. My first set actually had 0 contamination, which that was because Dr. Foster was looking over me the whole time. My second set had about a 1.5% contamination rate, and my third set had a 3.2% contamination rate. You're thinking, Ty, why is that number a little high? It's 3 plants. 3 plants got contaminated in 156 total transfers. So I'd consider that pretty successful. I was rather proud of that. And that allowed me to know that my technique was working and that we can move on to Bluebells, which is going to be my primary work next semester, and this afternoon was working with tissue from Bluebells that are emerging. Overall, there is a viability percentage here that is sort of neither here nor there. It's essentially anything above a 3 rating on there, which was fair tissue, so it wasn't contaminated, and it was starting to root at least a little. So we had a roughly 60% viability rating, but ultimately only time can tell what is going to be viable and what's not. So whatever ends up growing and multiplying is viable, and whatever doesn't isn't. Except for the contaminated ones. The contaminated ones are definitely not viable. So what's next? We're going to have stage 1 transfers, which is what we did this afternoon. That involves a further complicated process with bleach and tween, 20, and a bunch of other things that are keeping it clean. The big problem with stage 1 transfers is we don't want to kill the Bluebells. And if we kill the Bluebells, then our research is for nothing, right? So with bleach and with the tweed 20, we have to be very careful not to use too much or for too long, or else we will kill the tissue that we're transferring. More stage 0 preparation next week. Dr. Foster and I are going to go out to the other bed and move more into the Percival chambers, so that hopefully by the end of next semester we'll have another round to work with. And then variable control of stage 1 transfers. This is sort of the meat of what I'm doing, the meat of my research. And that is looking at things like, did we put it into cold? Did we force it through the Percival chambers? Was it grown in light? Was it not? Because we've found that the etiolated tissue has been really interesting to us. Etiolated tissue being that which was grown in the dark. And then seeing if preformed flower buds have anything to do with it. So we're using 3 separate groups. One uses at least a year old. One uses 1 to 2 years old, and one uses 3 years old when we know there have been buds on that plant. So that will be next semester and what we'll look into then. So in conclusion, I learned the technique. I succeeded there. That was the biggest part of my research this semester. The stage 0 Bluebells were prepared, and the initiation of primary tissue culture was delayed as of the creation of this PowerPoint, but we did do some today. So it hasn't been delayed too far, and we're just working on our technique with that. Again, we're trying to get that 1.9% contamination rate down to maybe 0.1%, something really low, which is something to strive for. So, any questions?